comes science. They might be giants, here comes science.
Hey, Dr. H here. Today's video will be our review of Unit 2 for the AP Biology test, where we will go over cell structure and function. Okay, here in part 1, we'll go over some basics of cell structure and very quickly go over, review some of the important organelles found in the cell. And then in part 2, we will get into cell membrane structure and function and get into cell transport. Okay, so let's go ahead and get started with part 1. We'll start here with some basics on cell structure. Of course, as you know, cells are very small, right? And the reason that they are small is to help with the exchange of materials with the environment, right? Both materials coming into the cell and materials coming out. And this small size helps with this exchange because of the very high surface area to volume ratio. Okay, small cells have a volume or a area to volume ratio of about 6:1. If we increase that size to a larger cell, then you see that the surface area to volume ratio there falls down to about 1.2. So that very small ratio there with the large cells would not work well to keep cells alive and efficiently exchanging materials with the environment. On the AP formula sheet, you will have all of these formulas in terms of surface area of different shapes and volumes of different shapes. I have seen a few example questions using these formulas, but again, if you're given the formulas and you have all the numbers and you have the calculator, you certainly should be able to plug all the numbers in. To remember that they are often looking for this surface area to volume ratio, right? So then you would just divide one by the other. Another way that cells will try to maximize this surface area to volume ratio is by greatly expanding their surface area with some structural features, like little tiny projections, like the cilia here, or the microvilli. These might be in the small intestine. And these little projections, they don't add a whole lot of volume to the cell, but they greatly expand the surface area. Right? So any cells that are along a surface where exchange is going to be happening, we would certainly expect to see some sort of structural feature like this to increase that surface area and maximize that exchange with the environment. The next general topic with regard to cell structure is this idea of compartmentalization. Okay, and this basically just means that inside of a cell there are different regions that perform specific tasks, right? And certainly when we think about complex cells like eukaryotic cells, we see that there is a whole lot of compartmentalization here, and we'll certainly talk a little bit more about this when we get into membranes and organelles. But we also see this compartmentalization in the simple bacterial cells, right? Even though there are no internal membranes, no internal organelles here, there are still regions where certain aspects of their life will occur, right? Like the nucleoid region where the DNA is contained, right? They don't have a nucleus, right, with a membrane around it, but they do have a region where the DNA will be concentrated. Okay, so this idea of compartmentalization, kind of separating out all of these different functions of the cell, is very important, again, to maximize the efficiency. So that's our basics of cell structure. Really not a whole lot there. Cell, we're just really just looking very basically at cells. So the next thing we want to talk about here are some of the important organelles. Now, remember, these organelles are just small internal structures of the cell that perform specific molecular biochemical roles. The only organelle that is found in all domains of life, right? Bacteria, archaea, and eukaryotes are the ribosomes, right? All the rest of these are all membrane bound, right? So they're surrounded by a membrane. So those would only be found in eukaryotes. Okay, and you can see around here the list of 7 organelles that the AP course lists as being important. I'm not going to go into detail on all of these. I'm going to run through them really quick and just say a few words about them. There are a couple here, though, that we will see again as we go through specific cellular processes, and I will certainly highlight those as we go. So first off, we have the ribosome. Right, this is the site of protein synthesis. Obviously, we will see this again when we get to the process of translation, right, making proteins. And remember, this is the only organelle that is found in all domains of life, right? Bacteria, archaea, and eukaryotes. The endoplasmic reticulum, 2 types here, right? Smooth ER and rough ER. The rough endoplasmic reticulum has ribosomes around the outside, and we'll see the rough ER again when we get to protein production, because it is involved in sending proteins out of the cell or moving proteins around the cell to the right location, right? So we will see this one again when we get to that point. The Golgi complex kind of works with the rough ER in sending proteins out to where they belong. Not really going to see this one again too much. Lysosome, this is involved in digestion, right? In some unicellular eukaryotes, the lysosome may be their entire digestive system, right? They bring in food particles, transfer those into a lysosome, and the digestive enzymes inside will break those food particles down. They're also used to recycle organelles, right? As organelles wear down, they are sent to the lysosome, and they're broken down again into their molecular components and can then be reused. See the vacuole. This is just a large kind of a central storage area. It's especially important, especially prominent in plant cells, where it will fill up with water and will actually put pressure on the cell walls of the plant, and that pressure is called turgor pressure. Okay, and that is what actually allows plants to stand up straight, right, against gravity. They don't have a skeleton to hold them up as many animals do, so they rely on this vacuole being full of water and pushing out on the cell wall to maintain that upright. The next, the mitochondrion. So this is an important one. Again, we will see this one. This will come back in Unit 3 about energy. So we will certainly talk much more about it then. So this is where a lot of the aerobic reactions of cellular respiration will occur. Okay, and we'll talk about a lot of these structural features here and some of these specific locations in Unit 3 with the mitochondrion. And we'll also hit in Unit 3 the other big energy organelle, the chloroplast, right? This is the site of photosynthesis, right? So again, we'll hit all of these structural features and some of these specific reactions that happen in each of those areas when we get to Unit 3. Okay, so those are the organelles that you should be somewhat aware of. Very rarely have I seen questions that are just straight up asking about one of these organelles. Like, there's not going to be like a big matching section where they give you a function and you have to match the organelle to it. Really, the ones that come up again later are the ones that you should really be aware of, and those we will definitely cover when we get to them, when we go over future content, right? Like the ribosome, mitochondria, chloroplast, maybe the rough ER in terms of moving proteins around the cell. Other than that, I think those are the main important ones. Okay, we'll now go over cell membrane structure and function. Okay, and with the function part, we'll mainly be talking about cellular transport. Okay, so first, just some general information about membranes. Of course, all cells are surrounded by membranes, right? This is true for all domains of life. And there are a few cell types, such as plants, fungi, bacteria, probably should throw archaea in there as well, that do have cell walls around the outside of the membrane. But we're not really going to talk about that too much right now. Okay, in terms of the major roles of these cell membranes, there are three major roles here. First, just very basically, they separate the inside from the outside, right? They form the boundary of the cell. They regulate transport, which is what we'll really be getting into in this video. And as we'll see in Unit 4, they also play a big role in communication. Looking at internal membranes, those of course are only found in eukaryotic cells. The main role of those is in compartmentalization, right? Something that I talked about back in part 1. But when we have all of these internal membranes here dividing the cell out into all of these organelles, that is one of the major roles, right? And again, this is to help increase the efficiency, right? Each different biochemical process has its own location, right? Bounded by a membrane. Okay, so let's talk a little bit about membrane structure, right? The current model of how cell membranes work is called the fluid mosaic model. And this is made up of two pieces, right? The fluid portion of the membrane is the phospholipids, right? Kind of shown here in white and orange. And then the mosaic portion is all of the proteins, right? And those are sort of the purple pieces in the membrane diagram here, right? So we'll talk about each of these structural features, starting with the phospholipids, right? As the name implies, these are lipids, right? And remember that lipids generally are insoluble in water, right? They have the big, long fatty acid tails, which make them very hydrophobic. Phospholipids are a little unique in that they have a polar region, right? They have what are called a polar head, which makes that region hydrophilic. Means that it can form hydrogen bonds with water because of these two different properties, right? The hydrophilic polar head group and the hydrophobic nonpolar tails. When phospholipids are mixed with water, they will spontaneously form this bilayer structure, right? With the heads facing out towards the water and the tails sandwiched in the middle, pushing all the water away, right? So this is the bilayer structure that we see in cell membranes, right? And this will spontaneously form, meaning that this is very energetically stable, right? This is the lowest energy form. A few different structural features to talk about with the different phospholipids that we may find in the cell membrane. First are the hydrocarbon tails, the fatty acid tails. Are they unsaturated or saturated? Remember, unsaturated means that there will be some carbon-carbon double bonds. And that introduces a little bend, right? A little kink in that fatty acid tail. That plays a very important role in maintaining the fluid state of the membrane at colder temperatures. Remember, that little kink will prevent the phospholipid molecules from packing too tightly together. So as it gets cold, and molecules tend to stop moving around so much, tend to pack in a little bit, these unsaturated fatty acids will maintain the membrane's fluidity. Organisms that are adapted to living in cold environments would tend to have more of these unsaturated fatty acids in their membranes. Cholesterol there also is found in some animal cell membranes, and that sort of works as a kind of a moderation molecule, I guess. It keeps the membrane from getting too liquid and from getting too viscous, right? So it kind of keeps it right in the middle, right? Kind of like the it's like a little Goldilocks molecule, I guess, right? It keeps the membrane fluidity right where it should be. So that's the phospholipid portion. The protein portion, that's the mosaic part of the fluid mosaic model. And the proteins are all over the membrane. And one important structural aspect of these proteins to think about is if they if they span the membrane, they're going to have different surface characteristics on these different regions of the protein. So if we look, for example, at a protein here that is going through the membrane, right, the portions of the membrane that are exposed either to the outside, the extracellular side of the membrane, or on the inside of the cell, the cytoplasmic side, since those are going to be exposed to this aqueous solution, those regions would be hydrophilic or polar. The regions in the middle that are sandwiched in between, that are embedded in the membrane, that are associating with those fatty acid tails, right? Those regions of the protein surface should be hydrophobic, right, or nonpolar, right, and they would interact well with that region. Okay, so that's membrane structure. So let's get into a little bit about membrane function. Okay, the main function that we're going to talk about here with regard to the membranes is transport. Okay, regulating what gets in and what gets out of a cell. Okay, and this is mainly due to the phospholipid portion of the membrane. Right, this hydrophobic polar region in the middle of the membrane there is going to prevent any polar molecule, any large molecule, from moving into the cell easily. There certainly are some molecules, like for example oxygen, or that can dissolve very easily through the bilayer. Right, any dissolved gas should be small enough to sort of fit right through. Right, and this is called simple diffusion. And we will talk more about this, the different types of transport, in a few minutes. The protein portion of the membrane has many different roles, as you would expect from proteins. Right, in general, proteins have lots of different jobs. And we see a few of those roles here. Again, in this video for this unit, the main thing we're going to be talking about with regard to proteins are the transport proteins, right? Which I think is in the upper left-hand corner of the diagram there. All right, so let's talk about transport. Before we get into cell transport, we really need to talk about a few kind of more general terms, mainly diffusion. Right? Diffusion, I have the definition written over there. It's basically just the movement of substances from an area of high concentration to an area of low concentration. Right? So we call this substances moving down the concentration gradient. Okay? And it's very important that we understand this idea of concentration gradient, right? Because it's going to come back in a few different units. We will see this idea of concentration gradients, right? And it is simply just a difference in concentration of some substance in two different areas. And what's going to happen if things are able to freely diffuse, then they will move from high to low until equilibrium is reached, right? And at equilibrium, there will be equal concentrations of the substance on both areas, right? On throughout the whole area. At equilibrium, that does not mean that molecules stop moving, right? They are still moving around. Just there is no net movement from one area to another. One way that we can look at this in a laboratory setting is with the use of this piece of equipment called a U-tube, right? And it is a piece of glass there, shaped like a U, and down at the bottom there will be a membrane, right? And in this case, it is a membrane, a selectively permeable membrane that is going to let some molecules through and other molecules not, right? In this case, we see that this membrane is letting water through, but not the large dissolved sugar molecules. So what's going to happen? The water is going to move, but the sugar is not. And it's the water is going to try to reach equilibrium. Okay, so in this case, the water is going to be moving from left to right. And that movement of water, it's really just the diffusion of water, is called osmosis. Okay, and there's nothing really special about osmosis. It is just the diffusion of water from an area of high concentration to an area of low concentration, right? And we usually don't think about water as having a concentration, right? But there are some ways that we can think about it that we will go over in a few slides. All right, so looking at cellular transport, the first type of transport that we'll talk about here is passive transport. Okay, and as the name implies, passive. This requires no energy input from the cell. Okay, these are molecules that are moving by diffusion, right? Moving down the concentration gradient from an area of high concentration to an area of low concentration. Okay, and there are a few different types of passive transport. There is this simple diffusion, right? And these are this is the small nonpolar molecules that are able to fit right through the membrane, right? You can see the oxygen gas there kind of squeezing right in between the molecules of the phospholipid bilayer. Hey, so dissolved gases move very easily through this type of transport, right? Simple diffusion. Larger molecules or polar or charged molecules are not going to be able to move through the membrane like this, right? Either they are too big to squeeze between the phospholipids, or if they're charged or polar, they're not going to be able to get through that nonpolar hydrophobic region of the membrane. So for those molecules, there is a process called facilitated diffusion, which, again, this is completely passive, right? No energy from the cell. But now there is a protein that is involved, right? Either a channel protein, which literally just has a hole through the middle for a specific solute to move through, or a carrier protein that will change shape a little bit and allow this solute to move into or out of a cell. Right? Both of these diagrams show things moving in, but diffusion can certainly work to move things out of a cell as well. Okay, but again, there is no energy input from the cell for any of these, any of this diffusion, any of these passive transports, right? Simple diffusion or facilitated diffusion as shown here. One other kind of thing to touch on here. This is the idea of tonicity and osmoregulation. Okay, like I said, when we think about osmosis, right, the direction that osmosis will move, the direction that water is going to diffuse, will go from high concentration to low concentration. And we usually don't think of water as having a concentration. So instead of looking at osmosis in terms of water concentration, we can think of it in terms of water potential. Okay, and the water will osmose from high potential to low potential. Okay, and there are a couple formulas here to calculate the water potential of a sample. Okay, and you see the formulas here, where water potential is the sum of two half potentials, right? The pressure potential and the solute potential. And there is the second formula there is how to calculate the solute potential. Again, with every formula that I'm giving you here, these will be on the formula sheet that you get with the test. So do not feel like you need to memorize these. I have seen questions in using these formulas show up on the test in the past, but these are really pretty straightforward formulas, right? One is adding two things together. The other is multiplying, four things together, right? And you will be given all of that information if you need to calculate water potential or the solute potential, right? But remember, the usefulness of this is to determine what direction is water going to diffuse. Right? It's going to move from high potential to low potential. The other piece here, or the next piece that we should talk about, tonicity, is what happens to a cell when it is placed in different concentrations of solute. Okay? And we use these three terms here to talk about the relative concentration of solutes inside versus outside of a cell. Okay, so hypotonic, that means that there is a lower concentration of solute outside the cell when compared to inside. Right? Isotonic means that they are the same, so they would be at equilibrium. And hypertonic means that there is a higher concentration of solute outside of the cell. Okay, and we see with the arrows there what would be happening to the water. Right? In a hypotonic solution, water is going to be coming into the cell, right? Because the water potential will be higher outside. In a hypertonic solution, the water will be moving out and the cell will be drying up because the water potential is higher inside of the cell. Keep those three terms in mind and remember that they are relative, right? In this case, we're saying that the outside solution is hypotonic to the cytoplasm. Right? You can't just have a solution, a beaker of salt water, and say that this is hypotonic, right? It has to be hypotonic to something else, right? And generally, it's to the inside of a cell. One final thing here to talk about with this, and this is this little guy here. This is a freshwater paramecium. They really love to talk about this guy just because it's a great example of an evolutionary adaptation to a specific environment. Right? So, like I said, this is a freshwater paramecium, so it will live in a hypotonic environment. So water is constantly going to be entering in to this cell. In order to survive, so it doesn't swell up and burst, these paramecium have the structure called a contractile vacuole. So this vacuole fills up with this excess water, and when it gets full, it contracts and removes the water. So this is a evolutionary adaptation that allows this paramecium to live in this hypotonic freshwater environment. Right? It's always important. It's always a good idea to kind of keep track of these evolutionary adaptations. Right? The College Board loves to throw in evolutionary connections whenever they can, right? So this is a really good evolutionary adaptation that has to do with cell transport. So that wraps up, I think, passive transport. So now let's take a look at active transport. Not surprisingly, this active transport is now going to require the input of energy from the cell, right? And the main type of energy that we're going to be using here will be ATP, right? That is the main energy molecule, right? And we'll certainly go over that in the Unit 3 video. So this is now moving substances against the concentration gradient, moving up the gradient, right? Going from an area of low concentration to an area of high concentration, right? And these are always going to involve some sort of protein, right? Usually they are referred to as pumps. To move substances against the gradient requires an active protein, right? They're not just going to do this on their own like we saw with passive transport. So the one most widely used example of active transport is the sodium-potassium pump. Okay, and here we see the steps of the sodium-potassium pump. Basically what is happening, 3 sodium ions bind to the pump from the inside of the cell, from the cytoplasm. ATP is used, and that causes the protein to change shape. The sodium ions are released to the outside of the cell, and now 2 potassium ions from the outside of the cell can bind to the other side of the protein, and the protein flips back to its original state, and the potassiums are released into the cell. Okay, so the overall effect of this sodium-potassium pump is to pump 3 sodium ions out of the cell and 2 potassium ions into the cell. Okay, so it is a 3-for-2 exchange, right? And we will talk about this sodium-potassium pump again briefly when we get into the cell communication unit, because this does form the basis for our nervous system. So let's talk about a few different types of active transport that are important. First one here is called co-transport. Okay, and this is when there is a concentration gradient produced through active transport, and that gradient is then used to transport something else. Okay, and this kind of introduces this concept of a concentration gradient having energy. Okay, and energy is sometimes very tough to conceptualize, because you can't really show a picture of energy, right? You can't draw a diagram of energy. You can't point to energy and say, there it is. It's just, a thing that systems have, right? And just remember that whenever there is a gradient set up, whenever there is a concentration gradient across a membrane, there is energy in that gradient. And co-transport is a way for cells to use that energy gradient to move something else into the cell. So here is an example of a co-transport system. There are 2 proteins involved, right? The proton pump there, that is the actual active transport protein, right? And you see it is using ATP to pump hydrogen ions out of the cell, right? And create this hydrogen ion gradient, where there is high hydrogen concentration outside of the cell and a lower hydrogen concentration inside of the cell, right? So now that there is this gradient set up across the cell membrane, there is energy here. The cell can now use that energy in the gradient through this other protein, the hydrogen sucrose cotransporter, which will allow hydrogens to diffuse back into the cell, right? Hydrogens normally don't diffuse very well through the membrane, right, because they're charged, but this cotransporter allows the hydrogens to diffuse in, and as they diffuse in down the gradient, right, releasing that energy, the sucrose molecule, the sugar molecule, is pulled along with it, right? And we see the sucrose there moving from low outside of the cell to high inside of the cell, right? So the proton pump sets up the gradient, puts energy in, right? That energy comes from ATP initially and then is found in the gradient. And then the co-transporter protein uses that energy, allows hydrogen to diffuse back in, back down the gradient. That releases the energy and pulls the sucrose along with it. Okay, so this idea of co-transport is very important, used to move a lot of nutrients, right, in the small intestine, right? Not just sucrose, but a lot of the amino acids, a lot of the other sugars are pulled into our cells through this same type of mechanism. I think the final thing that we need to talk about here with active transport is what we call bulk transport. Okay, so this would be endocytosis or exocytosis, right, depending on which way things are moving, right? Endocytosis will be bringing things into the cell. Exocytosis is moving things out, right? And what happens here, I think I have some pictures, yep. For phagocytosis or pinocytosis, that is bringing in samples. So the membrane just sort of moves around and forms the little vesicle and brings in pieces, right? In phagocytosis, that's bringing in a large solid piece, right? So it says there a food particle, or it could be another cell. In pinocytosis, it's really just bringing in a little sample of the extracellular fluid. For receptor-mediated endocytosis, there are receptors on the cell surface which are specifically binding to a particular solute out in the exterior of the cell, and those come in and sort of increase the concentration of that one particular solute in the little vesicle there, right? So receptor-mediated endocytosis is a little bit more specific than pinocytosis, which just takes a random sample. So that is the end there of transport. So that wraps up the Unit 2 review video. So I hope that all made sense, and good luck on your test.
Dropping science like Galileo dropped the orange.